Hello everyone. We all have come across words like chromosome, DNA, gene in our lower standards. But what are they made up of? Where are they located? Let's try to answer all these queries in today's session. We all know living organisms are made up of cell. In the center of the cell, we have the nucleus. Now let's zoom in more to see what's inside the nucleus. Inside the nucleus, we see spherical structures called as nucleolus. Can you see these thread-like structures? Do you know what are they called? They are called as chromatin fibers. What if the cell has to undergo cell division? Is it possible to divide such a cell into two equal halves with such a tangled bundle of threads? Of course not. So just before cell division, these chromatin fibers condense to form chromosomes. These chromatin fibers just before cell division condense to give you structure called as chromosomes. Now the next question is, what are these chromosomes made up of? If you zoom into the structure of the chromosome, we come to know that they are made up of DNA. Each chromosome is made up of DNA. The full form of DNA is deoxyribonucleic acid. The functional unit of DNA is called as a gene. Is the gene further made up of something? Yes, it is made up of series of nucleotides. Now what is a nucleotide? A nucleotide is a chemical structure which is made up of three units. Number 1, pentose sugar. Number 2, nitrogen base. Number 3, phosphate molecule. So we have pentose sugar, nitrogen base and phosphate molecule joined together to form a nucleotide. Now let's study about the nucleotide in detail. One of the components of the nucleotide is a nitrogen base. Nitrogen base in a DNA is of four types. First, adenine. Second, thymine. Third, cytosine. And fourth, guanine. Adenine and thymine together form purines. Cytosine and guanine together form pyrimidenes. Let's study about the DNA in detail. As we can see that the DNA is a double-stranded structure. It is also called as a double helix structure. If you want to imagine how a DNA looks like, imagine a circular staircase. If we open up this DNA molecule, this is how it would appear. The handles of the staircase are made up of alternate molecules of sugar and phosphate, whereas the stairs on which we climb are made up of nitrogen bases which are bonded together by the Chargaff's rule. What is the Chargaff's rule? Chargaff's rule says that adenine will only pair up with thymine by 2 hydrogen bonds. Similarly, guanine will only pair up with cytosine with 3 hydrogen bonds. We have now understood the structure and location of genes, DNA and chromosome inside a cell. Let us now understand their function. Genes define the biological characters of an individual. Characters like height, color of the skin, type of hair can be called as biological characters. For these characters to be seen in an individual, we require proteins. Proteins are a series of amino acids joined to one another. For these amino acids to be manufactured in the cell, we require codons. Codons are a group of 3 nucleotides present on the DNA. Let us understand it like this. Various such codons help us to manufacture different types of amino acids. Different types of amino acids joined together in a particular sequence will make different types of proteins. Different types of these proteins give rise to different biological characters seen in an individual. This completes our first session on basics of genetics. Thank you for watching.